So this is the second set of slides about the cell structure. We went over the first set of slides in class, just going over the organelles of the cell. This will be due on Tuesday when you return, because you do not have school on Monday. So some other organelles that you need to know, and I would just kind of keep a running chart of these. When we talk about plant cells specifically, plant cells have these things called plastids. And so we have to backtrack a little bit. When we talk about chromoplasts are responsible for photosynthesis. They are the specific parts of our chloroplast that do the photosynthetic work. Anything that has plast in them has to do with storage. Typically, leucoplasts are responsible for the storage of starch specifically. Chloroplasts are going to be storing the energy that is used for photosynthesis. So plastids is basically a group of organelles that strictly does storage. So again, plastids are going to be storage organelles. Specifically, chromoplasts are in plant cells. Leucoplasts are going to be mostly in animal cells. When we talk about the chloroplast as opposed to the chloroplast has three outer membranes. The two outer membranes are smooth, and the inner one makes stacks called thylakoids or grana. The chlorophyll and other pigments are found in the thylakoid membrane, which is where the light energy is converted to chemical energy. And we can see images of it above. Remember that the endosymbiosis hypothesis is why we get these double membrane cells. The idea that it was actually enveloped by an original cell, which makes it a double membrane cell, which is how we get eukaryotic cells from a single-celled organism way back when we only had single-celled organisms. So when we break down the chloroplast a little bit further, the chloroplast is an organelle in plant cells. It has stacks of thylakoids. In a in a stack of thylakoids, we call them a granum of thylakoids. The matrix found around the thylakoid is called the stroma. It contains enzymes and carbohydrates, or enzymes for the carbohydrate synthesis. A single disc in the stack of granum is called a thylakoid. Other organelles that we need to know, ribosomes are the workbenches, or basically the organelle that does protein synthesis. We've talked about these organelles before. They are made from rRNA, obviously ribosomal RNA, and they are two proteins that are two subunits that form this larger protein, which is the ribosome. Not only is it a protein, it makes proteins. They do not contain any membranes. They are just these proteins, and cells have thousands of these because we are making proteins all the time. If you remember, the mRNA runs through the large subunit and the small subunit, and it reads the codons, and it creates the amino acid sequence. Oops. Again, when we talk about the evolution of organelles, we think back to the endosymbiosis hypothesis. The endomembrane system includes all the organelles that are derived from one another, or which are continuous with one another. They have evolved from the folding in of plasma membrane over time. The membrane system includes the nucleus, the ER, the Golgi apparatus, and lysosomes. Basically anything that was originally part of the outer plasma membrane. And so we see that cells became more complex as the outer membrane started to have folds, and then those folds pinched off and created organelles within the cell. Very similar to the endosymbiosis hypothesis. This is an example of that. We see the yellow plasma membrane. When it gets this little convolution, eventually it can pinch off and become its own vesicle. And then through evolution, we can actually get it becoming its own organelle completely. This is again looking at how that evolution happens. We start with just a plasma membrane, cytoplasm, and DNA in the center. What ends up starting to happen is we have some inversions in the plasma membrane, and it starts making some of the organelles. The first organelles that we get are the nuclear envelope, which surrounds the DNA, and then the nucleus, which surrounds that, and then the next thing that we get is the endoplasmic reticulum. From there, we might have the cell envelop another prokaryote, which is part of the endosymbiosis hypothesis, which is how we get like a mitochondria or a chloroplast. That's going to be a heterotrophic prokaryote for the mitochondria, which does cellular respiration, or a photosynthetic prokaryote, which does photosynthesis. Those eventually evolve into either mitochondria or plastids and then chloroplasts, and we get our very first ancestral eukaryotic cells. The cytoskeleton, considered an organelle itself, gives the cell shape, provides movement of organelles, and we see something called cytoplasmic streaming in plant cells, which we could talk about. When you zoom in, we see there are tons of microtubules and microfilaments. These are proteins that are support specifically for the cell. We cannot see this, but basically the microtubules provide a track similar to a train on the tracks for things to move through the cell. If this was moving, you can look this up on YouTube. You can see the things moving through the cell. These microtubules are proteins. They are made of tubulin and a vesicle, or something that needs to be transported through the cell, basically attaches to a motor protein, and ATP powers this little motor protein, and it runs down the length of the tubule to get to where it needs to be. The tubulin also makes flagella and cilia. They are also made up of microtubules. Tubules. These are the things that propel cells. Remember, flagella are the long tails. Cilia are going to be just one, cilia are going to be all sorts of little kind of finger-like projections that move in waving motions to propel something in a direction. And we see that here. Ciliated cells, we can also see that here. Paramecium with cilia, sperm cells with tails, all sorts of stuff. Both flagella and cilia are covered with a plasma membrane, so the microtubules are enveloped by this plasma membrane. The main difference is the motion. We see that in the flagella, we see a swishing back and forth motion. With the cilia, they're going to move in a very different way to propel something forward still, but the movements are different. So microtubules and centrioles are organizing centers for animal cells. Basically, centrioles, right, if we remember this is part of mitosis and meiosis, are made of microtubules, which are made of tubulin. There are 9 microtubules in a centriole, and these, if you recall, go to the ends of the cells to start making the microtubules that pull the chromosomes apart at the center of mitosis and meiosis. We also have this thing called basal bodies, which anchor cilia and flagella, which are also made by microtubules. So you might want to make a kind of like microtubule column with all the things that it makes. Microfilaments, on the other hand, are made from 2 intertwined strands of actin subunits. They are much thinner and they have slightly different purposes as well. If you recall, microtubules are a tubulin dimer, which is 2 tubulin strands, proteins, wound together. So, for example, obviously we know actin is part of the muscle contractions and cytoplasmic streaming, which is the movement of an amoebic cell, so something without hands and feet and legs and things like that. It also has to do with cytoplasmic streaming. The cytoplasm of a plant cell actually like flows around the nucleus consistently, and the way that they do this is the microfilaments move in like a sweeping motion to kind of create like a whirlpool, similar to like if you've been in a lake and you and your friends run around in a circle to make the center of the water kind of move around like a toilet. It does the same thing. There are images of it. Just look up cytoplasmic streaming in plant cells. There are some intermediate filaments as well, and they basically maintain cell shape. They provide anchorage for other organelles, so they're not like floating around the inside of the cell. They are super coiled into really thick cables. They are usually keratin proteins. Most cells have material external of the plasma membrane. Cell walls are found in prokaryotes, plants, fungi, and some protists. Cell walls of plants are made of cellulose, and fungi, they are made of chitin. In prokaryotes, it's muramic acid, and in protists, they vary a lot. Animal cells are the cells that do not have cell walls. If you remember, we have a cell membrane. Again, the best way to remember this is that walls are often very rigid or more solid, whereas our cell membranes are a little bit more flexible. The plasmodesmata are channels between plant cells that allow the direct flow of the cell's cytoplasm to adjacent cells. So when we talked about the cytoplasm that moves and flows, this is why, because then it can access multiple cells. And so the plasmodesmata are these little channels. They're rigid. They're just openings. Think about a doorway without a door where the fluid can flow. Animal cells lack cell walls, but they have what's called an extracellular matrix. This is made of glycoproteins, such as collagen, proteoglycan, fibronectins. These glycoproteins are connected to receptor proteins in the cell membrane. They are used for support, adhesion, movement, and identity. Essentially, what this is saying is that that because we do not have a cell wall, we have other evolutionary structures that allow us to still protect and let things in and out, specifically these things. Animal cell junctions are going to look different, or the connections between animal cells. While plants only have plasmodesmata, we have three types. The three types are tight junctions, where the cells are pressed together. It's cell-to-cell contact. Desmosomes, or anchoring junctions, fasten them together in really strong sheets. And then gap junctions provide cytoplasmic channels between adjacent cells. They're just like plasmodesmata. So some cells are just touching, some are fastened for a specific purpose, and some have a channel that allows stuff to move across. Feel free to look up the Bioflix if you would like to. Again, there are both. If you look up Bioflix, you can see the tour of the plant cell or the tour of the animal cell. They are both on YouTube.